Welcome to the fascinating world of soil microbiology. Beneath our feet lies a hidden world, teeming with billions of microorganisms that sustain life on Earth. In this video, we'll explore how the groundbreaking work of three scientists laid the foundation for our understanding of this hidden world. Martinus Beijerinck, Sergei Winogradsky, and Selman Waksman revolutionized how we view soil and its vital role in supporting life. These pioneers developed revolutionary techniques and concepts that remain fundamental to soil science today. Their discoveries opened the door to understanding the complex relationships between soil microorganisms and their environment. Let's now explore the specific contributions of each of these pioneering scientists. Martinus Beijerinck, who lived from 1851 to 1931, revolutionized microbiology with his innovative research techniques. His most significant contribution was the development of the enrichment culture technique, a cornerstone method for isolating specific bacteria from complex environments like soil. This technique works by creating conditions that favor the growth of specific microorganisms while inhibiting others. As the process continues, the target microbes become more concentrated. Using his enrichment culture technique, Beijerinck made several groundbreaking discoveries in microbiology. He identified Azotobacter, a free-living bacterium capable of fixing atmospheric nitrogen into forms plants can use. Beijerinck discovered Rhizobium, bacteria that form symbiotic relationships with legume roots, creating nodules for nitrogen fixation. He also identified Lactobacillus, bacteria essential for fermentation processes. Perhaps most significantly, Beijerinck confirmed that the tobacco mosaic disease was caused by a new type of pathogen, a virus, marking the first identification of viral pathogens. Beijerinck's pioneering work opened the door to understanding the diverse microbial life in soil and established foundational techniques that microbiologists still use today. Next, we delve into the contributions of Sergei Winogradsky, a Russian microbiologist who lived from 1856 to 1953. Winogradsky made several groundbreaking discoveries that fundamentally changed our understanding of soil microorganisms and their ecological roles. Winogradsky discovered the biological process of nitrification, a crucial step in the nitrogen cycle. He showed that nitrification occurs in two steps, each performed by different bacteria. Winogradsky pioneered the concept of biogeochemical cycles, elucidating the microorganisms involved in the nitrogen, carbon, and sulfur cycles. In the nitrogen cycle, he identified how different bacteria transform nitrogen between its various forms. He also investigated the carbon cycle, showing how microbes decompose organic matter and recycle carbon compounds. Additionally, Winogradsky studied the sulfur cycle, discovering bacteria that could oxidize sulfur compounds for energy. Winogradsky demonstrated two important nutritional strategies in soil bacteria, autotrophy and chemolithotrophy. He showed that autotrophic bacteria can make their own food using carbon dioxide as a carbon source, similar to how plants perform photosynthesis. More remarkably, he discovered chemolithotrophic bacteria that derive energy from inorganic compounds like hydrogen sulfide, iron, or sulfur, rather than from organic matter. One of Winogradsky's most significant discoveries was Clostridium pastorianum, an anaerobic nitrogen-fixing bacterium. This bacterium lives in oxygen-free environments and can convert atmospheric nitrogen into ammonia, a form plants can use. This discovery was crucial for understanding how nitrogen naturally enters soil systems, enriching soil fertility without human intervention. Winogradsky's research fundamentally changed our understanding of soil ecology by revealing how microbes drive essential biogeochemical processes in soil. Now, let's explore the work of Selman Waksman, whose contributions to soil microbiology led to revolutionary medical discoveries. Born in 1888 in Ukraine, Waksman pursued his education in microbiology, eventually earning his PhD from the University of California, Berkeley in 1915. In 1939, Waksman coined the term antibiotics to describe substances produced by microorganisms that inhibit the growth of other microorganisms. His most significant discovery came in 1945 when his lab identified streptomycin, the first effective antibiotic against tuberculosis. For his groundbreaking work, Waxman was awarded the Nobel Prize in Physiology or Medicine in 1952. His legacy continued until his passing in 1973. Waxman's key contributions to science were numerous and impactful. Beyond coining the term antibiotics, he led a systematic approach to screening soil microbes for antimicrobial properties. His lab discovered over 20 antibiotics, including streptomycin, neomycin, and actinomycin. This pioneering work earned him the Nobel Prize in 1952. The discovery of streptomycin in 1945 was particularly significant. Isolated from the soil bacterium Streptomyces griseus, it was the first effective treatment for tuberculosis, a disease that had plagued humanity for centuries. Streptomycin reduced tuberculosis mortality by more than 50% and highlighted the enormous potential of soil microorganisms as sources of life-saving medicines. Waxman's work revolutionized medicine by establishing soil as a reservoir for drug discovery. His systematic approaches to antibiotic screening created a blueprint that researchers still follow today. He founded the Institute of Microbiology at Rutgers University, creating a center dedicated to exploring the medical applications of soil microorganisms. Waxman's pioneering work demonstrated that soil microbiology had practical applications far beyond agriculture, establishing a new frontier in pharmaceutical research that continues to yield life-saving treatments today. Modern soil microbiology is experiencing a renaissance thanks to advanced technologies. Central to this revolution are omics technologies, powerful analytical tools that provide unprecedented insights into soil microbial communities. Genomics studies DNA sequences while proteomics analyzes proteins. Transcriptomics examines RNA transcripts, and metabolomics investigates biochemical processes. High-throughput sequencing technologies are revolutionizing our ability to analyze microbial DNA in soil samples. These techniques generate millions of DNA sequence reads that can be assembled to identify the vast diversity of microorganisms present in soil. Microbiome analysis allows scientists to comprehensively study all microorganisms in a soil sample, revealing complex interactions and functions. These advanced technologies are driving applications in soil health assessment, crop yield improvement, and disease suppression. By providing unprecedented detail about soil microbial communities, these technologies are helping us understand and improve soil ecosystems in ways the pioneers of soil microbiology could only dream of. Soil microbes are incredibly diverse and serve multiple critical functions in the soil ecosystem. The soil contains various microorganisms including bacteria, archaea, fungi, algae, protozoa, nematodes, and viruses. These microorganisms perform 5 vital functions essential for healthy soils and ecosystems. First, microbes break down organic matter like dead plants and animals, converting them into simpler compounds. Second, they're crucial for nutrient cycling, transforming elements like nitrogen and phosphorus into forms plants can use. Third, certain microbes can detoxify harmful chemicals through bioremediation, cleaning polluted soils. Fourth, beneficial microbes suppress soil-borne diseases by competing with or directly attacking pathogens. Finally, microorganisms help create soil structure, forming aggregates that improve water retention and root growth. One of the most important zones of microbial activity is the rhizosphere, the area surrounding plant roots. The rhizosphere is a hotspot of microbial activity where roots release sugars and organic compounds that attract and feed microorganisms. In summary, soil microbes are essential for ecosystem function, plant health, and sustainable agriculture. Understanding these microscopic organisms gives us insights into how to manage and protect our soils. The importance of microbial diversity cannot be overstated in soil health. Soils with a high diversity of microorganisms tend to be healthier and more resilient to stress factors. Different microorganisms create a balanced ecosystem where each species contributes unique functions to the soil. Soil enzymes, primarily produced by bacteria, fungi, and plant roots, are critical catalysts for many soil processes. These enzymes influence soil structure stabilization, break down organic matter, facilitate nutrient cycling, and help degrade pollutants. Furthermore, soil microbes play a key role in carbon sequestration. They determine whether carbon is stored in the soil as organic matter or released back into the atmosphere as carbon dioxide. Ultimately, a thriving microbial community is essential for a healthy planet, affecting everything from plant growth to climate regulation. By preserving and enhancing soil microbial diversity, we support the foundation of terrestrial ecosystems. The future of soil microbiology lies in innovative approaches that combine sustainability with cutting-edge technology. Interdisciplinary collaboration is essential for advancing the field. When microbiologists work with chemists, ecologists, and agricultural scientists, we gain a more comprehensive understanding of soil ecosystems. Long-term studies spanning decades are crucial to observe changes in soil health and microbial communities over time. These studies provide valuable data on how agricultural practices affect soil ecosystems. Microbial inoculants are emerging as sustainable alternatives to chemical fertilizers and pesticides. These formulations contain beneficial microorganisms that enhance plant growth while maintaining soil health. The combined use of omics technologies like genomics, proteomics, metabolomics, and transcriptomics allows researchers to comprehensively analyze soil microbiomes. These advanced techniques reveal the genetic potential and functional activities of soil communities. High-throughput phenotyping combined with omics approaches helps us understand complex plant-soil microbe interactions. These technologies reveal how microorganisms influence plant growth and how plants, in turn, shape their microbial communities. As we advance our understanding through these innovative approaches, we can develop improved soil conservation methods, sustainable agricultural practices, and better management of terrestrial ecosystems. Our first case study takes us to Liberty State Park in New Jersey, a post-industrial urban area with significant soil metal contamination. Researchers examined soil metal contamination gradients to understand how microbial communities respond in these challenging environments. They discovered specialized microbial communities that could thrive despite high metal concentrations, providing valuable insights for phytoremediation strategies. Our second case study takes us to Sweden, where an agricultural consultant transformed a market garden using biologically complete compost. By introducing compost rich in diverse microorganisms, the consultant was able to achieve remarkable results. The results were impressive: an average yield increase of 72% across 8 different crops. This dramatic improvement was primarily due to enhanced soil microbial diversity and improved nutrient cycling in the soil. Our final case study examines the Whiting Farm, which faced both degraded soil and financial challenges. The farm implemented a soil food web approach, focusing on restoring the complete ecosystem of soil organisms. By fostering diverse communities of bacteria, fungi, protozoa, and nematodes, they restored the biological balance in their soil. This comprehensive approach not only restored their land, but also helped them achieve financial stability through improved crop production. These real-world examples clearly demonstrate how understanding soil microbiology leads to practical solutions for environmental and agricultural challenges. By restoring microbial communities, we can reverse soil degradation and create healthy soil ecosystems that support sustainable agriculture. The pioneering work of Beyerink, Winogradsky, and Waxman continues to inspire soil microbiologists today. Their discoveries have transformed our understanding of the microbial world within soil, from enrichment cultures to nutrient cycles to antibiotic discovery. Their work has provided practical solutions in 3 key areas: sustainable agriculture, with biological fertilizers and crop protection methods derived from soil microorganisms; environmental remediation, where microbial processes help decontaminate and restore damaged soils; and human health, through antibiotics and pharmaceutical compounds discovered in soil bacteria. By continuing to build on their foundation, we can unlock even more secrets of the soil microbial world. The study of soil microbiology remains as vital today as when these pioneers first began their work, holding keys to solving many of our planet's most pressing challenges.